Thanks. Let's continue. I'm the one separating you from the break, I guess. It's really a pleasure to have you here, Pauline. So I started with helic profiling of glycans in 2004. Actually, I went to Pauline's lab in Oxford and I got training for a couple of days. So I'm very grateful, and we have benefited from your work ever since. So if we look at glycosylation, I mean, Pauline's talk looked into different tissues, different cell types, different molecules. Gordon was focusing on plasma, I would say largely. And just to remind you, we are complex. We are complex organisms, humans with hundreds, thousands of cell types in different tissues, which respond to stimuli, to developmental stimuli, are genetically regulated and change with disease. So the glycomic complexity of a human is tremendous. And we virtually know nothing, if I may say so. So we know something, but really understanding how are we diverse, what is changing at the molecular levels, what are these glycans doing? I mean, Gordon was referring to the idea that glycans bind to lectins. That is indeed a paradigm which is still valid, but they do so many more things. They may play a role in our evolutionary makeup and so on. So I still think we know nothing about glycosylation. But I will still focus for a big part in my talk also on plasma, as Gordon did. We use a slightly different platform, and I want to introduce this in a few slides. So what we do is mass spectrometry. So we look at the very same glycans, not with fluorescent labeled CE or H+C separation, but in this case, what we do is we first release the glycans by PNGSF, we derivatize the sialic acid, and this is an important aspect which is coming back, because the sialic acids are the terminal parts on glycans, and those modulate a lot of interactions and are changing a lot with different physiological conditions. So what we do is we have two linkages here, a 2,3 linkage, which we turn into a lung tone, and a 2,6 linkage, which we turn into an ethyl ester. Simple chemistry, but by that we can simply, on the basis of the mass of a glycan, differentiate these isomers. We do a cleanup, we spot them on the MALDI target plate, and we measure it by MALDI mass spectrometry, and have our own software suite to profile hundreds of thousands of spectra in a day. This is the type of spectra we would get. So we resolve 80 or 100 glycans based on their mass. And we simply quantify the peak height and do a relative quantification or absolute one. And then we get these quantities of individual glycan peaks. And we then start calculating ratios. And that is very much linked to also what Pauline has presented, what also Gordon's lab is doing. So what we define then is glycan ratios which reflect biosynthesis. So we say, okay, we have this antenarity, and now there a fucose is being transferred, so let's take the ratio of the substrate and the product, and by that you have a glycan trait which reflects to a certain extent biological activity of the enzyme, of the enzymatic step. Then I'll look now into a few diseases, but before we move into diseases, just to recall what already Gordon and Pauline mentioned, we have age and sex dependence of glycosylation. But that's only a part of the dependencies. We have genetic dependencies. So if we look at disease changes of glycosylation, you always have this background of many other factors which change kind of the ground level from where you are starting. So that means if you use glycomics to measure disease changes, it's very strong if you do longitudinal changes, because you define your own baseline, and from there you see how a change in disease is affecting glycosylation. So we are different in glycosylation not only because of disease, but because of many, many factors. I will now have a few slides on pancreatic cancer, and the reason why I want to present the data on this disease is because the data we got are very consistent, which is not always the case. So we indeed have this replication problem, many of us have, and the studies we were doing were sizable but not huge, 100, 150 patients, sometimes 50 patients. So here we only had 45 cases, but sizable control groups. This is a study we published this year with the Mayo Clinics. And in red you see the disease, and then we have the controls, so not only healthy controls, but also people with pancreatitis, so relevant clinical controls. And you see that in certain glycan features are different. Not black and white, obviously, but they are different. And what you can do is then you can build a trait using 3 of these glycan traits and do a combined trait, and that's now indicated in red here. So you can compare the cancerous with the disease control or the healthy controls. And then what we have here in green is how our glycans perform. In blue is how CA19-9 performs, which is the established clinical marker. So you could say our marker is slightly worse than CA19-9. The nice thing is if you combine the two, they are highly orthogonal. So you go from areas under the curve of 0.85 to 0.95. So you get from a good marker to a very good marker by combining these two. But now the nice thing is this is not a one-hit wonder. I will show you 2 more studies where we find the very same signature. So the signature is defined here by these 8 traits, which we measured in the Mayo Clinic cohort, but which we also measured years before in our in-house cohort from the 80s. So we had historic samples, and if you look, we have the very same signature, the very same 6 glycans, and they go in the very same direction. So highly conserved signature. And now the nice thing is, I mean, these are sporadic cases of pancreatic cancer. We also have high-risk populations which have risk mutations. And those are monitored on a yearly basis, roughly. And that's what I show down here. So the numbers are low, but the very same changes we see in the cross-sectional study for the sporadic cases, we also see in these high-risk populations down here. So, and we see those before the disease event, before the formal diagnosis. I mean, Gordon mentioned it. These diseases are building up at the molecular level, and for pancreatic cancer, we know we are often late, very late with the diagnosis. So we think we touched on a marker which is generic for pancreatic cancer and can pick up also disease before the formal diagnosis, so can lead to an early diagnosis in these pancreatic cancer cases. And we're now replicating another time in these high-risk populations. So that is the one disease which I wanted to touch on, pancreatic cancer. And we went towards defining disease signatures in this way. So we calculated Cohen's d coefficients. So those show how is in this cross-sectional study the diseased group different from the control group. And we just gave it a heat map for all these glycosylation traits. So we tried to be a bit restrictive, not to have too many traits in there. But then you get kind of a heat map or a heat signature for the disease. And you see the 2 here are rather similar, not fully identical, but similar, clustering closely together. But now I'll go into fibrotic fatty liver disease, which is a pandemic. I don't know whether you are aware of this, but it's a big pandemic which is hitting us massively. And it's again linked to our lifestyle. But the point is, just looking at people, you can't tell who will have a fatty liver which is turning into a fibrotic liver. So there's a need for molecular markers to pick up this disease, and I will just have a few slides indicating how glycans can come in here. So this is the disease course. Your liver might get fatty, inflamed, fibrotic by fibroblasts coming in, and then you can move into cirrhosis and hepatocellular cancer. What we did is we used our glycomic technology and said, how are these fibrotic fatty liver disease patients different from healthy controls? And we found these glycan markers here. So what we see is the sialic acid linkage. The one thing our technique ideally picks up because of the derivatization is the one thing changing here. Because we saw that the 2,3 linkage is going down and the 2,6 linkage is going up. So glycans are not changing except for the sialic acid linkage out there. That's the oversimplification. And if we now look, according to fibrotic stages, which the pathologists define based on liver biopsies. So there we see that it's indeed already early stages. The first stage of liver fibrosis is picked up by this change in this marker, and the area under the curve is decent. So we have a good marker, way better than anything out there in the clinics now for early detection of liver fibrosis. And there's now medications coming onto the market. Some have hit the market, others are soon to hit the market. And we think this is something we would need to guide clinicians decide on who needs lifestyle changes, but maybe also medication to reverse disease course and prevent this catastrophic course of liver injury. And just back into this heat map here. So what we see is indeed the 2,3 linkage of sialic acids on dientenary, trientenary, tetranary glycans is going down. And we see that the 2,6 linkage is going up. So this is the one signature we pick up indeed for fibrotic fatty liver disease. But we also see it in type 2 diabetics. Why do we see it there? Because those are the patients at high risk of developing fibrotic fatty liver. So we pick up the very same patients also in type 2 diabetes. And we also see a similar signature in pancreatic cancer. So this makes us think, are we picking up a fibrosis signature? Because pancreatic cancer comes with a lot of stroma formation fibrosis. So we also now want to look in a more methodological way, maybe into lung fibrosis. Do we have a generic fibrotic signature across diseases? And I think this is what I want to do in the coming years. Look into disease signatures and look how glycosylation is really changing. Now, I mean, we miss the protein specificity here. We just look at released glycans, but already this tells us that not every disease is the same. So we have some level of commonalities, but also specificities of the disease-associated changes of the glycome. We can measure this with high precision. We can measure also these things from plasma serum, but also from dried blood spots. So that's quite nice. It gives the chance of maybe developing simple tests similar to glycan H. And I think there's really a massive need for clinical tests which can measure glycosylation. And these glycan profiles are robust with storage. Also, Gordon's team looked into that, so you can store dried blood spots, but also serum plasma samples for a long time without any notifiable change of the glycosylation signature. Very different from metabolic signatures, let's say. Okay. And now the second part of my talk is on antibody glycosylation, and actually I will try to move more into what is causing changes on antibody glycosylation. I'll have a few slides towards the end on that. So here we have the CH2 domain conserved glycosylation site with a diversity of glycans, which glycan H is covering and measuring. And those are functionally important. So we know, for example, that they change whether antibodies engage the 3A receptor, yes or no. And that's the fucose down here. So when the fucose is present, antibodies are silent and are not engaging the 3A receptor, whilst when the fucose is absent, we see that those antibodies bind to the 3A receptor and induce cytotoxicity. So we have a NK cell mediated killing of a cancer cell via the antibody which lacks the fucose, whilst when the fucose is present, those antibodies do not bind to the 3A receptor on the NK cell and will not mediate killing of a cancer cell. Known for a long time, but just to be aware that antibodies really mediate some effector functions based on glycosylation. Now, we are looking a lot into specific antibody populations, and for that we use a technique we termed or coined glycolyzer. So we capture antibodies in a type of ELISA experiment, we elute them, we digest them into glycopeptides and measure then the CH2 domain glycosylation via this LC-MS platform. And we also can quantify antibodies by simply spiking in a heavy labeled antibody variant. So we know the quantity of this standard, which we include, and based on that we can quantify how many antibodies are now captured for a certain specificity from this patient sample. And we did this a lot in collaboration with many of you who are also here in the audience, also with a rheumatologist from Leiden. And actually, I thought about which cases will I highlight. And the first thing is we did this for a lot of viral diseases. You know, with gestobidazole from Sanquin, we looked antiviral antibodies. How are they glycosylated? And what we saw is actually that anti-CMV antibodies, but also anti-HIV antibodies, oftentimes have this low fucosylation. Whilst, for example, a parvovirus always comes with a high level of fucosylation for the antibodies. So our hypothesis was, and to a certain extent still is, that membranous antigens, like an envelope virus, induce low fucose antibodies, whilst presentation of an antigen in a particulate form, like for a non-envelope virus, induce high level of fucosylated antibodies. So we lack the molecular understanding, but we see that there are differences in some settings, like envelope viruses. We have the low fucose, also for COVID-19, but in other cases, a soluble antigen will almost always induce high level of fucose IgG responses. Now, I will touch on two examples where I go a bit more in detail. And the first is transplantation, kidney transplantation. It's a study we did 2 years ago or 3 years ago with Margie Ackerman. 3 years ago. So we did the very same thing. We captured, in this case, pathogenic antibodies, which are against the HLA epitope on the kidney of the donor. So we captured these antibodies from the recipient, and we did the glyco profile. And actually what we saw is, when we compared the specific anti-HLA antibodies to the bulk, they came with a diverse range of fucosylation. So some had a rather low fucosylation, but also bisection was different. So these anti-HLA antibodies have certain glycosylation features, and we found the low fucose particularly interesting. And I'll not show a lot of data on this, but our very small study indicates that indeed those recipients of a kidney which show low fucose anti-HLA antibodies are at risk of AMR, of antibody-mediated rejection. So this low fucosylation may be indeed a risk factor that you may lose your kidney or at least get complications post-transplantation. And we're now looking into this. Can this be a marker for risk stratification upfront or for monitoring patients post-transplantation, those things. But the numbers are still lower, I have to admit here. Now the last example I want to show is on malaria infection. So if you look at the different stages of malaria, there is one stage in the erythrocyte where the parasite is replicating in the erythrocyte but has antigens in the membrane of the red blood cell. So why are those antigens there? They are there for mediating attachment to the endothelium, because what the parasite wants to avoid is liver passages with clearance of the infected erythrocyte. So it just wants to sit on the epithelium and replicate in the erythrocyte. And for that, it uses adhesins for conventional endothelium, but also in the case of pregnancy, somehow this parasite recognizes, oh, my host is pregnant, and makes a new molecule which mediates adhesion to the placenta. Now, what our collaborators asked was how are these antibodies, because these antigens of the parasite lead to antibody responses, how are these glycosylated, and could this help with protection or containment of the disease? Because what we see is when people are getting older, they are less susceptible to malaria. It's mainly the small kids who are most susceptible. And what we see is when people get older, the glycosylation changes, and we have these low fucose antibodies against the parasitic antigens in the red blood cell. And we think these help to contain the disease, clear the erythrocytes via ADCC or phagocytosis. So these are, we hypothesize, protective antibodies with a low fucose, which induce effector functions, while those of the kids with a high fucose don't engage NK cells and other immune cells. Now, if you look at the pregnancy situation, we see that with each parity, and that's shown here, I didn't put a line in here, but we see that there's a drop in fucosylation with each parity. So the more pregnancies a woman undergoes, the lower this fucosylation of these antibodies, and actually what is observed clinically, the better the mother contains again the malaria infection. So we see associations, no causality, but we think that also here fucosylation of the antibodies might be very important. Now these antigens, this one here, has been developed as a vaccine candidate, used as a soluble antigen, and it induces fully fucosylated antibodies. So we see the natural ones lack the fucose and are protective, and I would bet that tuning glycosylation with vaccination could help improving vaccine efficacy. That's our hypothesis. How am I doing? I'm out of time.
I was late.
So I have 2 more slides. Maybe 3. So this is our hypothesis in a way that antibody fucosylation is a double-edged sword. It can be protective, like in the case of malaria. That's our hypothesis. It can be devastating, like in the case of kidney transplantation and many other situations. And it would be great if we could steer this in certain directions. And that motivates the last few slides. We just had this paper coming out together with Mark Ehlers from Lübeck. So he does great work he's a mouse immunologist, I would say, also human immunologist. I can't tell this story, but I will just pick a few aspects of this story with Mark Ehlers. What he did, he asked, does an adjuvant change the glycosylation of antibodies? So can we, simply by choosing a proper adjuvant, have a higher or lower fucosylation of an antibody with vaccination? He used different adjuvants in a mouse model, and here he looked more into a cancer or malignancy model. So a cell which got the ova antigen, and what he did is he first immunized with ovalbumin, and by that he tried to get some protection against this artificial cancer, which homes to the lung. And what you see is this is a well-protected lung, so hardly any dark dots, but this is a very bad lung with many metastases. So this is a simple way to assess protection and efficacy of antibodies based on adjuvants. And what you saw is indeed it matters. So certain adjuvants lead to protection, hardly any metastases. Others lead to dark lungs, so no protection against this in this cancer model. So the number of lung metastases is the readout. Now, is this somehow linked to glycosylation? What Mark did is he looked not only at the lungs, but he also collected serum of plasma at the end when the animals were sacrificed, and we looked at the anti-ova antibodies, and we saw there's some afucosylation in some of these antibodies, but only for those mice which were protected. So we saw a nice correlation between protection against tumor formation in the lung and low fucosylation. I mean, it's not really very low, but it's a difference. So here all antibodies are fucosylated, and here 1% is not fucosylated. And now the interesting thing is this protection is conveyed by serum transfer. So if you simply transfer serum from a vaccinated animal, you can convey some of this protection. But if you knock out NK cell, you lose the protection. So it gives some insights. It's slightly antibodies, and they may engage NK cells in preventing this lung metastases. And this is just to show you how these profiles look like. We mainly have fucosylated glycan structures on these antibodies, but indeed some of them, like down here, some low abundant species lack the fucose, and we hypothesize that those are the antibodies which are then able to engage NK cells and kill the cancerous cells in this animal model. Now the last slide I have is on another experiment. So this was simply an experiment where we said, how do adjuvants induce different types of glycosylation on antibodies? And I won't go through the entire slide, but here I want to show, here we have the different adjuvants, and we see the higher this fucosylation, the lower the lung metastases, depending on the different adjuvants. And that's again highlighted here. So what are the factors of the antibody properties which associate with low metastases? And that's mainly the afucosylation of IgG1. So that's a major factor which associates with protection from metastases. And now I'll just summarize shortly. I looked first with you into the plasma and glycome. I think it's a rich reservoir of biomarkers. Glycans change in different directions with different diseases, and we looked into pancreatic cancer, where I see potential for the early detection in high-risk populations. We looked at a liver fibrosis marker for fatty liver disease, which we hope can help to pick up patients at risk and reverse, for example, the disease via lifestyle changes. And then we looked into this glycolyzer technology for looking at specific antibodies. Antibody afucosylation may be involved in the control of malaria infection. I showed you some data on that. And then we looked into AMR. And there, again, afucosylation is a risk factor of AMR. We have to work more on that. And then the mouse experiments indicate that we can induce afucosylation and that immunodeterminants like the use of an adjuvant matter for steering the immune reaction in a certain direction. And I hope we get more insights on that we can steer antibody glycosylation, and by that use this double-edged sword in a careful manner. Thanks for your attention.